Sweet, all right. If you're ready, I'll just start recording and we'll go into it.
Yep, go ahead.
Cool, so quick, 15 seconds. Who are you?
Okay.
And who are you, and what is your background in terms of the person reviewing this clip?
Oh, I'm Florian. Nice to meet you. I am a 47-year-old scientist who is working as a consultant and ghostwriter.
Nice. What's the best way for people to contact you?
People contact me is I guess via email, flurich@gmail.com. Yep.
Sounds good. All right, so let's just jump into it.
All right.
Let's pop open this video.
The Joe Rogan Experience.
So I wanted to talk to you about this
Whoops.
cloning.
All right, let's get this thing open properly. Awesome. All right, let me make it small.
The company trying to clone woolly mammoth.
Yes.
That's like Jurassic Park with sheep, right?
Pretty much. This guy's name is Forrest Galante, and he's like this, how do you even describe it? He's like a biologist, and he kind of like travels around the world looking for extinct animals or like rare animals. I think he does other things as well. But he basically talks about this company trying to clone woolly mammoths. So let's see if I listened to this and I'm like, I wonder if this is possible, like even possible. Like, let me get Florian's opinion on this. So let's see.
The Joe Rogan Experience.
So I wanted to talk to you about this cloning and the rewilding of the mammoths and all of that stuff. I'm going to Colossal tomorrow to learn a little bit more about it myself.
Explain Colossal.
Yeah, so Colossal Biosciences is this, if you ask me, incredible company. They are, by their own declaration, a de-extinction company.
We're a little out of sync. Like, I see the video, but the sound is like 0.3 seconds early or late.
Okay. Here, what I'm gonna do is, let me send you the link.
Okay.
And then, why don't you just, so I won't have it playing, or what I'll do is, why don't you just watch it?
Okay.
We can start from 0 again. Tell me when you hit play, and then when you hit pause, I'll hit pause also. And I'll just mute the sound.
Okay, 1 second. Okay. 3, 2, 1, go.
Nice.
All right. De-extinction. Oh yeah, that guy. Yeah, that guy is
Cellular scientist.
absolutely.
Specific.
Yeah, that guy is a leader. Yep.
Wait, which guy exactly? Let's pause it real quick.
George Church. He actually sounded the alarm when people wanted to try to, set out mosquitoes that are resistant against malaria. That idea that you put out mosquitoes into the wild, and then they take over with beneficial properties like genetically modified mosquitoes, and then they take over the genetically inferior populations, like you can extinct insects as a disease vector in that way, right? That's the thought behind it. But of course, if something goes wrong and you made a mistake you didn't realize, you can't put the genie back into the bottle, right? So once the mosquitoes are out, there's no way of reversing what you did. So he sounded the alarm about that.
But with the mosquitoes,
I have to look at this.
They wanted to with the mosquitoes, they wanted to introduce like a sterile version or something.
Yes.
Is that what it was?
Yes, I think something like that. That they had different versions, like genetically modified sterile, but I think they were not all sterile. There were also different ideas, but but researchers thought what should go wrong. It worked under controlled lab environment, so it will work in the outside world as well. And he called for an ethical committee to talk about that first. That was George Church. That was actually the first time I heard about George Church, and that was back 10 years ago. And George Church has been around for much, much longer. So I guess if that guy is on board, then you are somewhat ethically sound, I would say.
Okay. Just out of curiosity, what did he think could go wrong? Like, what did he say? Were some of the dangers?
I don't remember, but let me look it up again.
I mean, it's I could see that like I could see that going wrong. I can't really imagine necessarily how, but just in my gut, I'm like that could probably end badly. Like,
okay, yeah. So Harvard, George Church is at Harvard. Is a Harvard University geneticist. Okay, he said. Wait a second. That's different. Yeah, he has like several ideas on different ethical questions in science, and with that, even with someone like George Church, you have to also look at the background, who sponsors the guy, because if you have an eminent ethical authority, one way to co-opt his opinions, or her opinion, whoever it may be, is by simply paying them a lot of money, and then if you have them on your side, then you're boosting your public persona as well, right? And that can be done. Scientists can be had cheap, so you can let them give a talk in your foundation and get them on board. Scientists don't earn much, right? If you earn I think the top salaries for scientists that receive an NIH grant is topped off at $230,000 per year. That seems to be a lot if you grew up as a postdoc making $40,000 a year. But if you compare that to the salaries you get at companies, it's nothing, right? So that makes scientists unfortunately a little bribable. Not saying that George Church is, but that's just a general observation I have. People think it's always beneficial to, like, politicians shouldn't be paid so much, and public officials shouldn't get so much money. Yeah, I'm not sure though. I mean, you want to pay them well so that they don't get the temptation to take on any bribes, right? But let's go back to the George Church mosquitoes.
Yeah, speaking of those bribes, I think I was listening to like Nancy Pelosi got like some speaking fee of like $300,000 or something like that.
Yeah.
For an appearance.
Yeah. Of course, of course they do that. If you get like a person like Pelosi on board, then you can push around like any policy you want, pretty much, right? I mean, you can't overdo it, but you have like a lot of leeway if you have people like that on board. But there's also other committees where the politicians are not even well known, like who runs the Ways and Means Committee in Congress. Like, nobody knows. I should know. I don't know right now, but the people on that committee are the ones that are actually deciding which funds are going to be released. This is eminently an extremely important committee. If you have them on board, then you can push you can make sure that there are company-friendly policies implemented, or like that people don't even talk about questions that could be unpleasant for you as a company, right? So let me see. It takes a longer while to check what his point is. Here, mosquitoes. Right. No, I don't get walks. Gene drive. I think he isn't. I think he isn't especially against that from the get-go. He has more. He is more. He raised like genetic questions or ethical questions, and he wants to make sure that people adhere to those ethical questions. But I don't think he advocated for or against that method of gene drive in general, right? I mean, with CRISPR, I think he only called for moratorium against CRISPR to make sure you've talked about all ethical questions first before you pursue it further. But in the end, like one person who talks about ethical principles doesn't withstand once if it's not put into law, companies start doing it, and once you have the first drugs that are produced with something like CRISPR, or even something like a gene drive where you release genetically modified organisms into the wild, then once the genie is out of the bottle, and once you start healing people with that, nobody's going to listen to ethicists who say we should put a stop to these experiments. That's just how it normally goes, right? So you have basically companies that just start doing things without much concern for ethical questions, and then if it helps, like, everyone will be on board. It's just not possible to influence public opinion against gene drives or anything like that at that point.
So I guess the fear is, it's like, okay, we have regulation in the States, nobody can use CRISPR to modify humans. So then they're like, oh, we're just going to go to China. Like, is there anything you can do? Like, you know what I mean? Like, is it inevitable? Like, what can be done about this?
Well, I think eminently even China was scared by the prospect of changing human embryos, and they actually imprisoned the guy, right? That did that. There was a Chinese guy who did that. And interestingly, George Church was on board with that. Or George Church said it's okay to modify human embryos if you can eradicate certain diseases, right? I would take a different stance to that, because what you should do instead is you should select embryos before they are born, pre-implantation diagnostics, right? You would select select embryos instead of genetically modifying them. You would basically test embryos before they're implanted in the uterus for genetic defects, and then you just don't choose the ones that have those defects. Then you don't need to do any gene technology. Then you don't need to do any engineering, right? You just simply choose the embryos that are going to be healthy. But on the other hand, that of course would mean that we had to have a society where everything is where reproduction doesn't happen naturally anymore, which is an absolute nightmare. So I'm not entirely sure. I mean, pre-implantation diagnostic is done routinely, I think, or should be done on embryos of people that can't conceive naturally, right? So then they should select the embryos, whether they're healthy, like looking that they're healthy, right? That I think makes sense, right? There's no reason you should select an embryo that has some severe genetic disease. But on the other hand, that also opens up like a whole Pandora's box in and of itself. I mean, where do you put the limit, right? What is going to be acceptable for society and what not? Now society determines in the end how societal members should look like, and that's a big problem. You already have high schools in the Bronx, or not? Yeah, Bronx probably, Manhattan. You have some private schools in New York that start to demand genetic testing to get the most intelligent children.
Wow.
Right.
Private school?
That was, yeah, it was a high school. Yeah, it was a high school in Manhattan that did that 10 years or 5 years ago. And I don't think whether that was a trial run and authorities have shut it down, or whether it has caught on. There was a huge discussion about that. So because it's also not sure how efficient that is, right? You can probably select for some more for some students that are intelligent and that are especially athletic, but based on DNA, you're going to miss a lot. You're going to have a lot of false positives and false negatives. In other words, you're going to get, if you try selecting like that, you get rid of a lot of candidates that are actually really smart. You eliminate them because their DNA doesn't look like you expect, and you take a lot of candidates that you think will be intelligent but are actually not, right? Just based on their DNA pattern. And if you automate the process, I mean, you could basically, the problem is that many genetic traits, it's not like you have like one gene that has a mutation and then you don't have a certain trait. Many traits are polygenic, meaning that instead of like there's not a genetic defect isn't there because there's one gene that has a mutation, there's like one gene that is knocked out. That's not the case for many traits. For many traits, it's like, for example, if you take multiple sclerosis, there's not one mutation that's responsible for multiple sclerosis. It is rather like 1,000 genes not working as good as they could. And then the combination of that gives you multiple sclerosis symptoms. And now good luck finding those genes, right? If you look for genes that are a little bit changed, you find a lot of genes that are a little bit changed. Our DNA is not as homogeneous as we would think. There's a lot of genes that are like where the protein product is a little different, just a little bit, and you cannot decide whether that genetic product is really a problem or not, right? You cannot decide whether this little change makes the protein 1% less efficient. You don't know. And so then good luck sifting through a million of genes, or not a million, but sifting through a million base pairs and finding among like 70,000 genes, find the ones that are combined together give you multiple sclerosis. So that's impossible almost. Now good luck finding genes that if you combine them make people more intelligent. That's almost impossible. We don't even know how to define intelligence in the first place. You know, you could say, okay, that person is very smart with numbers, or that person is a very good salesman. That's obviously very, very smart people. But I mean,
right.
Can you really pinpoint it down to certain genes? Almost impossible. And if you let computers do that, they will probably do a better job, artificial intelligence. But then you don't understand what these computers decide, right? So it's still a black box to you what genes are deciding whether someone is more intelligent or less.
Okay, so, interesting point. So I guess going back to this video with the woolly mammoths, I'm curious to see what you think about what they say. Let's keep going.
Why did we stop in the first place?
You were talking about John something with the ethics.
George Church, yeah. Okay,
yes.
Let's just let's start again from zero.
From zero, okay.
Okay, let's start again. 3, 2, 1, go.
The Joe Rogan Experience.
So I wanted to talk to you about cloning and the rewilding.
You have it on as well. I hear echo now. We should just do it from your.
Let me see. Okay. Because I'm trying
Let's just do it from your thing.
Okay. I just wanted to listen to it so I could know what they're like. If you stopped and you addressed a point, then I wouldn't
the
be
mammoths
lost.
and
Oh,
all
should
that
I
stuff.
stop?
I'm going to.
Should I stop once I address a point? Should I stop and then do a comment, or should I just go ahead and
What do you
just
mean?
comment while the video is running?
I would pause the video so that we like keep our place, and then you talk,
Okay.
and then I'll just unpause it, and then we just keep going.
Okay. All right.
All right. Okay. Cool.
Colossal tomorrow to learn a little bit more about
Explain
it myself.
colossal.
Yeah, so colossal biosciences is this, if you ask me, incredible
Random question: have you heard of Colossal Biosciences?
No, not before.
company, and they are, by their own declaration, a de-extinction company. So it's this guy, Ben Lamb, and he's got George Church, who's a world-leading cellular scientist. I don't know the specifics, of de-extinction and cloning and CRISPR and so on and so forth. And they've come together and raised a ton of money, and they are de-extincting animals. And the science is there. Like, it's done. All it took was the money, basically, behind it. And they've put together this incredible Rolodex of scientists and people. And it's real-life Jurassic Park with purpose.
Where are they going to put them?
Yeah.
So there's a couple different things going on. So the first one they're working on is the woolly mammoth, right? And this isn't just for fun. This has real, like, important conservation implications, which is really fascinating.
Yeah, but you select which ones to conserve, which one you don't, right? I mean, okay, so there's many species going extinct every day. So which ones are important for your food chain, for example? Which ones are important? Which ones harbor other organisms that are also extinct? That was a topic they talked about Jurassic Park. Like, you have these dinosaurs, and then you have lots of microbes that live on top of those dinosaurs. You need to recreate them as well to have them be able to live in that environment. They somehow magically solved that in the movie and the book, but that would be one concern, right?
The microbes, you'd have to revive the microbes. Is that what you're saying?
Yeah, you would have to basically in a dinosaur. You can't just take a mammoth or a dinosaur or de-extinct. You can't just de-extinct creatures and put them into the wild. They will probably die, or there's a good possibility that they die because they don't have all the other members in there that they had back then in their habitat, right? So it's not there. Maybe they died off because some of the other species weren't there, right? So I guess you can try and then see where it leads you. I mean, you have invasive species all the time, right? Like fish get introduced into a different habitat or different ecosystem, and now all of a sudden they completely take over because they don't have any enemies there. And those fish survive, right? So I guess you can just I guess you can just select some animals, but it may not go smooth once you have them reintroduced into the wild.
Yeah. So I guess what you're saying is, like, if you revive a lion, then you need the gazelle also, because then he needs to eat something. Is that kind of the point?
That's true, and it may either be that the lion was relying on a gazelle, so that the lion dies after being reintroduced because there are no gazelles, or alternatively, the lion may discover other food sources and then just completely take over, right? That can both happen. Or, I mean, of course, maybe the lion just smoothly integrates into the whole ecosystem. That can also happen, right? But unintended consequences can go in both extremes.
That reminds me, actually, like, about the Yellowstone situation where they just killed all the wolves, all the local ranchers and people. So I think because they had livestock, so they were like, all right, well, I ain't trying to have wolves. So then after the wolves were killed, there was like some ecosystem imbalance where, like, the elk just started overgrazing and whatever. So it kind of like messed up everything, basically. It was like a chain reaction. And then they brought some wolves from Canada. I think they brought timber wolves. And then they released them into Yellowstone. And it seems like things are kind of like finding balance again. But it's not perfect, obviously. But do you know anything about that?
Yeah, I've heard about reintroducing wolves, and they have done that. I think not just Yellowstone Park. You have reintroduced wolves into regions of Europe as well. And I just read an article where local farmers were not happy about that. Of course, because those people were usually the ones who are calling for hunting all the wolves down. and for good reason, right? They have a commercial interest in that. But I think if you do that, if you just have wolves completely died out, and then in a certain ecosystem, and then you just reintroduce wolves from another source, yeah, that probably works. That can probably bring the balance back, right? Because it's like it is. Everything else is still there in the ecosystem, right? So if you just remove wolves for a few generations, then you bring them back, yeah, that's probably fine. There's probably not much changing. But even then, as you said, some things become different, right? So
Yeah, I think what
you
they
never
said.
restore the original balance.
Yeah, I think what they were saying with the timber wolves was that they brought the wolves from Canada, and like timber
Yeah.
wolves are generally like really big. Like they're like huge. They're like larger than they're like puma size, you know. So when they released them into the Yellowstone, like Yellowstone wolves originally were like not that big. They were like slightly bigger
Yeah.
than dogs. So now you got like these like mega wolves running around. It's kind of like it's not exactly the same. So
Well, that's true, but then they are from Canada, so you expect them to be polite.
yeah. Oh man. All right. Well, let's keep going here.
But they are.
Oh, sorry. On that last note, I guess with the conservation, like you could release the same species of wolf, I guess theoretically, instead of having some other weird variation.
Yeah.
They are planning on starting with, I think, 100 mammoths and putting them in this place called not Pleistocene Park, something like that. This park in Siberia that they've been doing this experiment on as to what happens when you add megafauna back into the Arctic tundra to offset carbon emissions.
Wow.
Offset carbon emissions.
They're using what DNA? Elephant DNA and mixing it with something else?
So it's Indian elephant is the closest living relative to the woolly mammoth.
What does an Indian elephant look like? Is it similar to an elephant?
Yeah, it's a smaller. So African elephants are bigger. They have the really big ears.
Right.
Indian elephants are typically the ones you'd see at the circus, with the pink in the ears, the smaller triangular shaped ears. So just a different species of elephant. And so they're taking Indian elephants and they're using CRISPR technology and they're using existing mammoth DNA and they're making an embryo and then they're implanting it into the Indian elephant, and 22 months later, an Indian elephant's gestation period, she will give birth to a mammoth.
A real mammoth.
A real mammoth.
So it's not...
Let me pause that right there. Is that, like, what's up with that? Is that possible?
Yeah, that's possible. Absolutely. I mean, you can modify the genome with CRISPR. You can very specifically cut some genes and insert whatever DNA you want. There's some unintended consequences always. So basically, the enzymes for CRISPR will also cut somewhere else. It's called off-target effects. So you got to be careful. If you just modify one gene, how it works is basically your CRISPR is like an enzymatic scissors that goes along your DNA and looks for a very, very specific sequence, right? Now you can calculate the odds. If you have you can have 4 base pairs in your DNA, adenine, guanine, cytosine, and thymidine, or ACGT with 1 letter code, and then finding either of these bases at one position, likelihood is 25%, and then finding 2, like a GC would be 25%, like finding a G would be 5%, 50%, GC 25%, then 12.5%, 6.25%, and so on and so forth, right? If you make the sequence long enough, then if you have a specific target sequence there, you make it long enough, you ensure that the enzyme only cuts there, but you always have some off-target effects because the enzymes, they may recognize some other sequences and mistake them. So you basically it's impossible to check for all off-target effects, right? So that's what you have to be careful about. But by and large, yeah, you can make it precise enough that you have a reasonable good chance of really in a very targeted manner do changes in the DNA that you want to do. Yeah, and then basically, you need some kind of mother womb to get the first mammoth, right? Because no mammoth exists. So you use an elephant. That's possible. Yeah, that sounds feasible so far. Yeah.
Sheesh. Okay. All right, let's keep going then.
Like a hybrid.
That's okay. That's a good point. So it is, in the sense of what they do is, if you imagine the DNA of an animal, right? And then you imagine the fragments that are broken out of it, right? What they're doing is they're taking that DNA of and I don't understand the cellular side of it very well. This is just my base level understanding of it. I can talk about the conservation side of it, but they're taking that double helix, that DNA, and all those pieces that are missing from the mammoth, they're putting in Indian elephant pieces. So you end up with an animal that is physically and morphologically identical to a mammoth, but has used all of the DNA from the closest living relatives in order to get there.
Phew. Boy. And this process, how long does this take?
That's good. That's accurate, pretty much.
Yeah, I mean that's what you would do, right? I wasn't thinking about that, but of course, I mean you don't. I mean it's probably not. It's probably not as easy as just bringing the entire mammoth back and just putting that into the womb of an elephant. There's probably some compatibility issues, and you need to do some modification. Also, I don't know how much mammoth DNA they could really retrieve, right? I mean, mammoth DNA is, I imagine that when you retrieve this old DNA, and DNA can actually survive for thousands of years. I mean, the way we would share DNA in the lab, like you have DNA to a specific gene, you want to share with a different lab. What you do is you spot the DNA from your solution. You take a drop of your solution, you put it on a spot of filter paper, and then you send out you let it dry, and you send out that filter paper by mail to another lab. So you would have you open the letter, and then you have a filter paper in there where someone drew like a circle with a pencil on, and then you cut out the circle, you put it into your solution, and then you have certain reagents that help you extract the DNA, and then you store it in your freezer. So DNA on this filter paper, sometimes you don't get immediately to taking the DNA back into your own fridge. So it can sit for 2-3 years on your bench, and then I forgot about this DNA. Let me extract the DNA from that filter paper, and it still works. So DNA can really survive for a long time. However, if you have mammoth DNA for 10,000 of years, there's bound to be some aberration happening, right? And this DNA is not neatly put on the filter paper either, right? So basically, you will have some damage to that mammoth DNA, I can imagine, right?
So, have you heard about where they're getting all these mammoths from?
No, I actually don't know where they're getting those mammoths from. But they're basically, I would imagine it's preserved mammoth, like that is frozen in ice.
Pretty much. It's literally like Siberia. Like as the ice is melting, like people are just going up there, and they're just finding like tons of mammoths, like thousands. And they found some like with the stomach contents still within the mammoth, like pretty crazy.
Nice.
Let me see if I can pull this up real quick. Tab. Let's see. Do you see this right here?
Yeah.
So I think that's like an example of one that they found. But these teams are basically.
Wait, that's the one they found? That's not a drawing. That's an actual mammoth they found.
Yeah, that's the actual thing. Let me see.
Oh, frack me! That's crazy.
Yeah, right. Did you know about this? Hold on.
No, I did not know about it. I didn't know how well they were preserved.
Right,
wow, remains. If I hear remains, I think there's some old spots of fur that they dig out somewhere. But
nah, they found the whole thing. They found the whole thing. This is
not the remains. This is a 10,000-year-old woolly mammoth.
Let's see. This one? Yes. Pulled from Siberian Lake. Here, remains of 10,000-year-old woolly man pulled from Siberian Lake. Let me.
I mean, that's good enough conserve. We're not talking about dinosaurs here. We're talking about. Yeah.
Let's see. Bottom of Siberian Lake. Spent 5 days scouring the sill of Lake Pechenelava in the remote Yamal Peninsula, which includes tendons, skin, and even excrement after they were spotted by local residents. About 90% of the animal has been retrieved during 2 expeditions. Here's the skeleton. They found the freaking baby, though. Let me see if I could... I guess that one's a different... But yeah, they pulled this one out, too. This one's pretty wild. Look at this. Here.
Yeah.
That thing's wild.
Poor baby.
30,000-year-olds in Yukon permafrost.
Yeah, that's cool.
So, okay, so you think this is good enough for the DNA? Like, what exactly? Let's say you literally pulled up this, right? Yeah. What do you need to do now in order to extract the DNA?
Okay, what I would need to do is I would cut out like a small patch of the skin, and then I basically put it into a solution in my lab that cracks up all the cellular membranes and that basically digests everything of the cell and sets the DNA free. So they're basically
like some kind of acid or something.
Say again?
Like some kind of acid or something?
Yeah, it's an acid cocktail. I forgot what it exactly was, but that's what you learn as a biochemist. Is basically one of the first things you learn is how to extract proteins and how to extract DNA. It's actually pretty cool. You can digest. I think we had digested liver at one point in my studies in like '97 when I was still at university studying biochemistry. We went to a local slaughterhouse, got like a beef liver. And then we would digest that beef liver, and would then use a glass rod. And at one point, when all the proteins and all the lipids were digested from the cells, and you had a solution where the DNA would swim around, you put like a glass rod in, and then you pull the glass rod out, and you turn the glass rod, and you basically get a long string of DNA that you can roll up on your glass rod. So that's pure DNA from that liver. And so you do this, a very similar thing probably with the mammoth. You probably need to be you probably need to use different enzymes, right? Because it's like, I mean, dissolving beef liver is different from dissolving like ancient mummified tissue, right? But it should be the same principle. But you also learn many things only by doing, because, for example, what we did back then in the lab, the first thing was we thought, hey, the protocol is kind of lengthy. Why do we wait 2 or 3 days to get everything digested while we have like this machine here that has a running blade? Let's just like grind the liver up with this like fast rotating blade, and then we can directly extract the DNA. And we did that, and we got no DNA. And the guy overseeing the lab was snickering because we were not the first ones like smart to do that, because he said, yeah, what you just did is you applied shearing forces. It's game over for the DNA, so you cannot do that. Yeah. Because DNA is a long string. If you take a rotating blade that cuts off everything, yeah, it will cut off everything, also your DNA, right? So that only illustrates that that's really learning by doing, right? You have to. It's basically at that level, biochemistry is very much an art, right? You have to learn how to treat the tissues, how to extract proteins or lipids or DNA. You can extract all that, but you really need to develop some solid techniques. So it's actually not that easy on the basic level, but you can do it, right? So there are protocols and people communicate that, and that's that. And then you extract the DNA.
Okay, so you basically put it in acid and then you extract like the string, right?
Yeah, you extract the string, and you have to basically the string was just as a demonstration what you can do. You basically have to if you want to extract certain genes, you can do that. But I guess if you want to then clone mammalian DNA, you need to have the cell nucleus intact, and you have to somehow find a way to put the nucleus of that mammoth cell into an elephant cell to make a hybrid, right? You need to have a clean. You basically need to take a oocyte that's just fertilized and replace that oocyte nucleus. Take that out and put your mammoth nucleus in.
Theoretically possible?
Yeah, and practically should be possible because you can do that with any cell. So you should also be doing that with mammoth. But I have to say, that's my first idea how to do it. I'm not a mammoth. I'm not an evolutionary biologist. I'm not a specialist how to do that kind of cloning. But that's something I would do. That's I mean, if you grind up the tissue and just isolate the DNA, then you can test for the presence of certain genes. That's what you can always do. If you want to have a livable DNA, you need to be more delicate and keep the nucleus with the DNA intact and put it into a different cell.
Okay. Well, okay, so going back to the acid and then you get the string, right?
Okay, yep.
First off, it's crazy that you can just have DNA like that. You can just see it in a string. That's crazy.
That is, yeah.
Secondly, so you have the string. So then, how do you start manipulating that? Like, as you said, CRISPR and stuff, right? How do you even? What is CRISPR exactly? Like, how do you get the strand into CRISPR?
So CRISPR is a methodology. So a lot of these, and it's basically it is basically a way to edit DNA. And so what you do is CRISPR originally is a bacterial defense mechanism, right? So bacteria are faced with a lot of threats from viruses, and so bacteria have developed a lot of enzymes to cut intruding DNA from viruses up, right? And also to edit DNA from viruses that enters the cells as a defense mechanism, right? You just edit the DNA of, like, you have this bacterial cell and new viral DNA comes in. You wanna either destroy the viral DNA or kind of render the DNA dysfunctional, right? You basically change the DNA. And researchers looked at that, and then they started thinking about, well, if bacteria can change DNA, then we can use those bacterial enzymes, isolate them, and use them to change other DNA. And at that point, it was very difficult to genetically manipulate some organisms, and it's a lengthy process where you classically in the 80s and 90s, what you would do if you want to clone a gene into, let's say, mouse to test a certain drug, you would basically take mouse DNA, have enzymes that cut out a string of DNA and then put new DNA in, and that took several generations. You have to grow up mice with that DNA. We extracted DNA from those mice, put them into other mice, and so on. It took like 3 years to get a genetically modified mouse. that's a bit too long, right? But that was the only way of doing it. And so with CRISPR, you now have the option to just directly manipulate the DNA in the cells, and that saves you several steps and makes the process much faster. So what CRISPR does is basically an enzyme or a combination of enzymes. One enzyme recognizes a certain target sequence, and you can design that enzyme so that it recognizes any target sequence you want. And so this enzyme.
How do you design an enzyme?
So basically, enzymes are proteins, right? And these proteins are basically, imagine a protein like a big sphere, and it has like an active center, something called an active center, where whatever reaction it does occurs. Like take trypsin for whatever, for example. Trypsin is an enzyme in your stomach, or pepsin. I think pepsin is the enzyme in your stomach, and it digests proteins. How does it do that? It recognizes proteins, it binds to them, and then it just hydrolyzes that protein, just cuts up that protein in its active center. And so if you want to design an enzyme that recognizes a specific DNA sequence, you just change that active center so that the amino acids in that active center, that the parts of the protein will actually start recognizing your specific DNA sequence, right? So you have a certain sequence of base pairs, let's say ACGG. Now your enzyme binds to the DNA and just rides back and forth on that DNA until it finds your target sequence, and then it attaches to that sequence. And then another part of the enzyme starts cutting that sequence and putting a third sequence in there, and then it closes the hole again.
That's crazy. So, that's insane.
Yeah, but it works like that. Imagine you have your target sequence. Your enzyme will now cut out, like let's say your target sequence is ACGG. It binds there, and then it changes the next base from a T to an A, right? So if you can see, actually,
that's crazy that they can get so specific.
Yeah, basically what you do is you what this does is you have your sequence, and then you basically swap out one base to the other. You bind your sequence, you swap out one base for another base, and then you let go again. And so that way you have changed one base pair. You can change more than one base pair, but you basically edit it directly in the DNA, right? You change the DNA at that point. And so you can basically, there's a lot of tricks and variations to that scientists have developed. And so you can, the point being is you can go take a DNA and basically design a certain base pair change that will change the protein that comes out of that base pair sequence in the end. That way, if you, for example, if you have a genetic defect, right? Maybe let's say your defect is caused by a mutation in one gene. You go and you change that mutation back to the healthy DNA part, and you eliminated that mutation. Your offspring will not have that disease anymore.
That's crazy. Well, okay, so I guess basically you so the enzyme that breaks up the DNA and makes the change, that's from bacteria that was studied messing with, it's like as like a counteractive agent to the viruses that were trying to change people's DNA, basically.
Yeah, if you're bacterium, and so what viruses do, they just invade a cell, and then they get that cell to reproduce the virus over and over and over again, right? And the cell obviously doesn't like that. If you're bacterium, you don't like someone doing that to you, so you develop a defense system that cuts up any incoming DNA. And so you just cut it off, threat is gone. And that's how many of these there are many different of these defense mechanisms of bacteria, and scientists have long used those enzymes that bacteria have for their own purpose, right? If you know that the bacteria has an enzyme that cuts up incoming DNA in a specific sequence, you just isolate the protein, and now you can use it to cut DNA and then cut any other DNA in the lab.
Okay, so I'm thinking of like the acid and then the string. I can like visualize that, right? But like with the enzymes and the bacteria, like that doesn't make sense to me basically. So like if you're a scientist and you're trying to extract the enzyme from the bacteria, how does that work? Like you just have like a petri dish with some culture in it with bacteria, and then how do you get the enzyme out?
Yeah. So what you do is you grow. First, you have a bacteria. You grow them. You grow them in culture, and then you take the bacteria. That like what you get the next day. You have so you start with like a little petri dish, and there you have like one colony. You take a little toothpick. You take a toothpick. You take the colony off there, scrape it off, and put it into a medium, into a growth medium that has a lot of like all the nutrients that the bacterium needs to grow. And then you put it into an incubator overnight that is at a temperature of 37 degrees, and that basically shakes all the time. And so you let it shake overnight. The next morning you come and you have like your culture is foggy because all the bacteria have grown in there. You take that culture and you take it, put it into centrifuge, and you let it spin really fast. And then after 20 minutes, your bacteria are in all the sediment. You take the sediment into a new buffer, and you basically crack open the cell wall of those bacteria, which then releases all the content of the bacteria into your medium. And then you basically isolate the protein that you get from that. There are certain solutions that you use that will basically dissolve the lipids, and then basically precipitate the proteins in there. And so then you spin that solution down again. All the lipids and other schlong that is in the solution you can pour out, and then you're left with sediment that's pure protein. And then you take that pure protein sediment into solution, put it into your freezer, and that's how you isolate a protein. In many cases, what people also do is they have a bacterium produce a certain protein for them. For example, insulin. You introduce DNA into a bacterium. That DNA instructs that bacterium now to produce insulin, and then you basically let the bacteria grow overnight, and let them secrete insulin into the medium, and then you basically take the medium and take the insulin from there. Basically, you use bacteria to grow whatever you want, and then you either isolate the protein from the bacteria, or you isolate the protein from the solution. Some proteins the bacteria will secrete into solution. Some proteins they grow inside. But there's always ways to basically isolate the protein from the bacteria solution that you have, and that's how you do it. That's a lot of biochemistry over the last since the 50s. People have developed so many different ways of like extracting all kinds of different proteins, and enzymes like CRISPR, the CRISPR enzymes. They are isolated in a certain similar way, and you use bacteria to grow them as well. I mean, this is just
okay. Quick question. So I figure out how to get the protein now. But the specific protein has to be programmed to the sequence, right? Is that how it goes?
So that you do by exactly. You program the protein before you do that, before you extract it. What you do is you basically you isolate the DNA that will produce those enzymes. I mean, you need to remember that the DNA sequence is translated into a specific protein sequence, right? And if you know the protein sequence, you can design a DNA that produces that specific protein, right? In theory, you could take a protein that's maybe 900 amino acids long, and then you design the entire DNA that codes for that enzyme. That's a little tedious because you would have to design 2,700 DNA bases. That takes too long. But what you can do is you can take the CRISPR protein sequence, or the CRISPR DNA sequence that will later on produce the CRISPR protein, and you take the DNA sequence and you look at the part of the DNA sequence that encodes for the active center of that enzyme, and you change it so that it produces amino acids that exclusively recognize a certain DNA target sequence that you want, right? So let's say amino acid like glycine. Let's take 3 amino acids. Let's say you have a CRISPR protein that recognizes the specific DNA sequence CAT, and let's say that CRISPR protein has 3 amino acids called alanine, glycine, and proline in its active center. And you can just change that by taking those amino acids, taking the DNA that encodes for those 3 amino acids inside the CRISPR protein DNA, and you just change it to 9 different base pairs. And then you introduce them into the lab. Now you have your modified CRISPR protein that recognizes your CAT target sequence. And so now you take that protein and you put it into a bacterium. You grow that bacterium overnight, and that bacterium will now produce your protein in masses. And then you isolate the protein from that bacterium, and voila, there you have your CRISPR protein.
Oh my God. Okay. Sheesh.
I mean, I'm actually, one thing that surprises me the most is that we haven't had like some disgruntled protein or DNA biochemist that just stays a little bit longer in the lab. They all stay, biologists all stay longer in the lab. They never have a 9-to-5 job. They always stay longer. It goes completely under the radar. If you want to create a designer bug in the lab, I don't want to scare anyone, but yeah, I'm surprised it hasn't happened yet. You can totally, I mean, there's zero limitations. If you want to develop like the new version of the plague, you could in theory do that, and nobody would realize you're doing that until it's too late. I mean, you have so many tools in the biological lab. You can change DNA of bacteria at will, within limits, right? It's not you cannot completely change it, but you can in theory you can in theory do the you can in theory weaponize COVID more if you wanted to. You can take the part that binds to the ACE receptor and put that part into the common flu, and then it's even more transmissible, or whatever you want, right? You can do whatever. I'm not as up to date in my viruses as I once was, but you can take whatever crazy virus you have, you can modify it so that it's now being transmissible like the flu. It's a theory. It's not a problem of doing that, and nobody would realize you're doing that until it's too late.
Well,
now you would need an infrastructure to deliver it, right? You cannot just you cannot just create a bug and then just like spray it outside of the lab. You need to have an infrastructure to get it among people. So it's not that easy, but in principle, you have a lot of tools to do whatever you like on the biological level.
That's wild. I guess people were talking about the lab leak hypothesis with regards to COVID, so maybe there is some truth to that. Maybe people were up to some weird stuff.
Yeah, some people are up to some weird stuff. Maybe some janitor was underpaid, as they probably all are in China. Of course, China doesn't admit it, but some janitor who is underpaid, who can't live from the happy tidings of socialism alone. So he decides to, instead of incinerating those bat cadavers that they gave him from the lab, he just sells them. Oh, this bat just died today. Well, I can just sell it on the wet market, and that's how COVID gets among people. That's the lab leak hypothesis. Or maybe somebody left, like somebody didn't like burn it, like the janitor or whoever was in charge of burning the cadavers. said, fuck it, I'm too late to my appointment. Instead of going to incinerate, let me just dump this into his trash bin here. And then like wild animal comes and discovers this, like eats the bats from the trash can, and then that animal gets sold. With if you have people that are lazy in terms of like keeping to the security protocols, that's also something that's not readily apparent, right? Somebody is lazy, doesn't observe the security precautions, things get out of control. That's not far-fetched, and that's not a conspiracy theory. That's a real problem that can happen.
Right? Well, okay, quick question. So going back to CRISPR, when people say CRISPR, I'm thinking of like a machine, right? But that's probably not the correct vision. Like you were talking about CRISPR enzymes and all this stuff. When people mention CRISPR, like what should people be visualizing?
I think people should visualize a molecular machine, like a micro machine. Micro machines latching on your DNA and just switching out base pairs, and then clearing mutations.
Well, a machine implies like some kind of metal, right? Like some kind of thing spewing out, like I don't know, like smoke or something, right?
Imagine computer game Pac-Man. Yeah. Imagine like this little Pac-Man latching on the DNA, moving along the DNA until it finds the sequence that is designed for, and just cutting out the bad base and putting a different base in. That's how people can. That's literally what happens.
So when people say, "Oh, we're going to use CRISPR," they're basically talking about the system to extract the bacteria and the protein, basically, and then the protein itself.
They're talking about the next step. They're talking about once you have the protein. They're talking about a protein that latches to the DNA and changes base pairs.
Okay. All right. Well, on that note, should we just continue with the video?
Yep. Go ahead.
Sweet. Okay. Over here. All right. Here we go.
So I think they've been going for about 5 years on the science, but the science of de-extinction and cloning, I mean, you remember Dolly the sheep, right? Mm-hmm. That was like a known thing. So that's been going on for a long time.
Yeah, Dolly the sheep, by the way, died.
Exactly.
Dolly the sheep, a lot of the problems with these cloned animals is that they are kind of sickly, right? They're not really healthy in many cases. And many of those, and if you know about Alien, I think it was the fourth part, where Ripley all of a sudden gets into this room where they have all these failed alien cloning experiments, like all these malformed fetuses. And yes, that happens. If you try to clone a mammal, you'll have many trials that are not working, and you end up with miserable failures. And that's really what happens. I mean, we actually had some in Hanover where I studied. There was a gallery. They kept some misformed, misshapen babies that died, for example, in a jar. Literally, they had them in jars and they showcased them. Like in the 20s and 30s, people would keep malformed babies, and they basically, if the parents agreed to have them serve science, then some of the researchers would put those malformed embryos into a jar and displayed them, and basically for educational purpose, right? But basically, when you clone a mammal, many things won't work. When we tried CRISPR in our lab, because we did that, every biology lab nowadays uses CRISPR. I just left biology when that was just coming up. Basically, what it looks like, it in the end, you envision it like a very smooth and easy way. What it looks like in practice is one guy in our lab who tried these different CRISPR versions. He had like 100 different petri dishes on his bench, and then he was actually injecting the DNA from each of those petri dishes, from each of those different bacterial cultures that had grown CRISPR enzyme. He all different combinations of DNA they were targeting, and he would inject all those different DNAs into zebrafish embryos and grow up those zebrafish. And then some of those zebrafish didn't have the proper mutation, and so you would end up growing a lot of animals and then selecting. It's a very inefficient selection process what you still have to go through, right? Until you once have your desired mutation, until you once have your desired organisms that doesn't have the mutation anymore, or whatever you wanted to try to do with CRISPR, you are going through a ton of failed organisms. It's just it.
Oh my god.
Yeah, I mean, cloning a mammal, it sounds like smooth, but I bet you those people went through months and months and years of failed attempts. In the end, there's some randomness to it, right? And you have to go through a lot of different experiments until you finally find your one mutation that worked out. Until you finally have your one mammoth DNA that you can really inject into an elephant cell or whatever you need, you use for reproduction. That's something that the public doesn't realize, but yeah.
It's starting to go from climate change, we're gonna bring back the animals, to just corpses, just failed. Oh my god.
I mean, yeah, corpses. It's not really. I mean,
just horrific
that's one
mutations.
side, right? Corpses, but
Dr.
for
Evil.
example, it's a practical thing. For example, we did a lot of let's say we have a certain zebrafish gene that we want to study. We did the research in zebrafish. It's the same for any organism, basically. Okay, so what we wanted to do was we wanted to introduce a certain mutation in a specific gene in zebrafish to study this gene by looking at the mutation of that gene. And so what happens is you harvest the eggs from zebrafish. That's a very simple process. And then you line them up in your pet, like a little dish, and you micro-inject them with your DNA. And you do 200, 300 embryos. You can do that in 10 minutes. Okay, then you grow, let's say you have 300 injected embryos. You grow them. Okay, so 20% will basically just die from the injection. Okay, so you are left with 80% that grow. Now, congratulations, you have 250 little zebrafish embryos that are now growing up, right? You have 250 different tanks. Imagine that. 6 months later, you go into the zebrafish facility and you have 250 different tanks. You need to test each and every single one of those zebrafish by crossing them into another, by crossing those with a non-mutant female or male. Now you have 250 families that you now have to test one by one which one of those has your mutation of choice that you introduce. And one problem with genetic experiments is often that if you have a mouse facility, that's a very expensive. It's a very, very expensive housing. A zebrafish facility, which is basically a huge room that has a certain climate and has a ability to house 500 different aquariums, for example. If you want to have a mouse house that houses 500 different mice families, basically, you have to have a exact temperature, exact humidity. You have to make it sterile so that there's no infections spreading in there. A mouse house costs $1 million to keep, and it costs a lot to maintain. So it's cost prohibitive in a way, right? That's also one reason why people wanted to develop CRISPR, because the easier you can introduce mutations, the better. But it's a lot of work and there's a lot of infrastructure involved, and it's not even creating monsters. It's just that that the mutation that you find, finally you have your mammoth DNA. To grow that up, you will have a lot of mammoth, a lot of elephant or whatever you create that will not carry your mammoth DNA to term. It's basically you have to do a lot of. You have your mammoth DNA. You put that into elephant cell. you have a lot of elephant cells that will simply not take, and then you grow them up. That's like that's a huge endeavor also, right? And not about creating monsters. It's just a huge infrastructural challenge what you need to overcome, right? So that's something that's not talked about, but that you immediately realize once you sit down and say, okay, let's clone a mammoth. Okay, so first we need to do this, and then we need to grow the elephant. God damn it. We need to grow 20 elephants. Yeah, good luck growing 20 elephants, or good luck having 20 pregnant elephants that have these mammoths in their womb. Yeah, I mean, think about that. That is a real problem, right? So you need to develop ways to pre-select successful experiments, right? So I'm just talking about that because those are things that nobody thinks about, that is not really privy to the inner machinations of these things. And that's basically what most of the research is like. If you try to clone mammoth, that 1% is actually cloning of the mammoth, 99% is problem solving, and most of those problems are infrastructural nature.
Interesting. Okay, quick question. So, any last comments on Dolly the sheep? Brought her up.
Yeah, Dolly the sheep. I didn't get much into it. I didn't look deeply into that. I can only talk about what I know from our zebrafish experiments, and I just bet you there were a lot of failed dollies that didn't work out as well. Yeah.
Yeah. What do you think it is about the process that gives you like a sickly animal? I mean, it seems kind of, I guess, obvious if you're just messing with people's DNA, trying to like splice stuff. But what do you think from your perspective? Like, why do you keep getting these sickly creatures?
Because what you're essentially trying to do is you put adult DNA that has already gone through some abrasion into a fresh stem cell. Telomeres are one problem. So basically, with each cell division, you basically reproduce your DNA, but each time you reproduce your DNA, a part of the ends cannot be efficiently reproduced. You always lose some of the end. So what DNA has are, if you have linear DNA, you got to start somewhere to multiply the DNA, right? And every time you start, you lose the last part of the DNA. And so DNA is something called telomeres. Telomeres are specific sequences at the end that are there as some sort of like a buffer that you're losing DNA that you can afford to lose at the end. It's basically some DNA attached to the end that can get lost every cell division. But at one point, your DNA cannot duplicate anymore, cannot replicate anymore, because you have these because it's just becoming too short. And so that's how we age. That's one process of our aging, right? Our DNA can only go through so many divisions. And if you put like a 40-year-old DNA into basically a new stem cell, then you basically have already a 40-year-old embryo when it's born. That was one problem with Dolly. And right, and you also have methylation patterns, some that we haven't well understood yet. So some genes are activated and switched on and switched off by attaching some modifications to it, right? Some methylation groups, for example. They put a very, very simple methyl group. There's a carbon atom with 3 hydrogen atoms on its end, and it's just attached to part of the DNA. And when an enzyme comes along that wants to switch on a certain gene, activate a certain gene, it sees there's a methyl group to that DNA, and so it stops activating it. And so over time, those those methylation patterns change in DNA. If you put if you took a methylated DNA back into a fresh cell, then that DNA isn't going to be fresh. that's already undergone that DNA that you put in layman's terms, that DNA has already been places, and you put that into a naive new egg cell. Yeah, that basically you get an embryo that's already somewhat aged. That's one problem.
Wow.
With this approach, right?
Wow. That makes sense. Yeah. Wow.
Right. Yeah.
So okay, so this is obviously not the same, but like, let's say you had an old person, say a 60-year-old guy with like 20-year-old woman, right? Is that like a similar kind of problem, or I mean, you know?
It may be, but usually since we know the 80-year-old men can have sex with 20-year-old women, certainly, and get health, and the resulting children are actually healthy, then it's possible. But there's one problem though, and we all know about that as well, right? The older you are when you conceive, the more likely it is that your offspring will have some defect, right? I mean, if women that conceive after the age of 35, for example, there's a much higher risk of trisomy 21, which is Down syndrome, and other genetic defects that can basically come up, that basically accumulate over time. And so, yeah, that is a problem in general, right?
Is that a telomere problem, like similar to the cloning, or is it
no? It's
separate?
It's not a telomere problem in that case, because your germ cells are producing fresh DNA, right? Your germ cells are producing DNA that's fresh from the start. So telomere shortage is not a problem if you are older when you conceive, but there's other mutations that are accumulating over time, and that just you know, the way oocytes are created by the body, and the way sperm cells are created by the body, that process of creation, that is prone to have some errors in there. For example, instead of like creating an egg cell that has that is divided, that has all chromosomes divided in two, right? So every cell in our body has two sets of chromosomes, but our germ cells only have one set of chromosomes, right? The mother produces one set of chromosomes and goes together with the father's one set of chromosome, and then your fertilized egg cell has again two sets of chromosomes. But the way you create these sperm cells and oocytes that have just one set of chromosomes, they are more error prone the older you are. So all of a sudden, you're not getting like a set that has like one set of chromosomes. You get some chromosomes that are that did not divide like 1 to 1, but 2 to 0, right? And now you create an embryo that has 2 chromosomes number 21 plus 1 chromosome number 21, and you get 3 chromosomes 21. That's Down syndrome, right? So, but of course, methylation patterns may also have an effect. I mean, a lot of this research has only been done in the last 10 to 20 years, or maybe 20 to 30 years, and yeah, sure, I don't want to rule that out.
Okay, cool, awesome. Well, let's keep going then.
Exactly. For like 20 grand, you can clone your dog. Yeah. So
it's kind of creepy.
It is. It's bizarre. But the point is, the science has been there for a while. There just hasn't really been the funding or the motivation for it. But what I think so fascinating, the reason I'm so like emotionally invested in it, is the conservation implications that it has. Because what this company is ultimately doing is rewilding species that humans have removed, and that's going to, in theory, in a lot of places, sort of fix the offset, the imbalance of the ecosystem.
Yeah, about that. Those animals have been removed for a good reason, right? Sometimes they have been inadvertently removed, but some of these animals, there was a reason that people removed them. So, just to keep in mind.
Quick question about the dog comment. You know anybody that cloned their dog?
No, I don't know, but I do know that you can buy glowfish, which basically are zebrafish that have a fluorescent protein in their genome. You know how these jellyfish sometimes like light up blue or green, like when the moonlight shines, you have all these like fluorescent colors in the ocean.
Same thing.
And those are jellyfish. Those jellyfish have a certain protein that's called green fluorescent protein. It's a very creative name because that's what it does. It has green fluorescent. And then once researchers discovered that, they discovered there's yellow fluorescent protein, there's red fluorescent protein. They discovered some corals have also fluorescent protein, and they isolated those proteins, and put them into zebrafish. And you can now buy zebrafish that glow in the dark from a pet shop. That's a genetically engineered organism. Not a dog, but a zebrafish.
Wow.
That's really interesting. There's a lot of debate about whether or not humans killed off the woolly mammoth, though, isn't there?
I think there is. Yeah, I think there is. And I can't really speak on that, but I do know that when the mammoths disappeared, so the Arctic used to be like the savannas of Africa. It used to be big grasslands, right? It wasn't all covered in trees and things. And that's a recent adaptation since the mammoths went away 10, 20,000 years ago. And so that's what's happened is the permafrost up there is melting pretty rapidly, right? Underneath that permafrost is like 1.5 trillion tons of carbon. And once that carbon enters the atmosphere, it heats things up like crazy. So by removing those mammoths, and I can explain why the mammoths keep it colder, but by removing those mammoths, it's allowing that permafrost to melt much quicker and release more carbon. So the idea from like a financial standpoint of how they make money is the carbon offset of putting mammoths back into the environment.
How do they make it colder?
So it's a couple different things. It's
real quick, with what he's saying about the carbon being under the ice, what is that? Carbon is like an organic material generally, right?
Yeah.
So when he's saying that carbon is going to be released into the atmosphere, like what exactly does that mean?
You must mean carbon dioxide, which is basically trapped in ice and then gets released in the atmosphere. I don't know. I mean, it sounds spectacular, but I'm not entirely sure how big of a threat that really is. Yeah, I guess maybe, but
well, how does it get trapped in ice? First of all, how does that work?
Like these gases are solved in water as well. You have oxygen solved in water. You have carbon dioxide solved in water as well. And when the water freezes, some of that will be actually trapped in ice.
Gotcha. Okay. Let's see what he's got to say.
Basically, when there's trees and shrubs, they take in more heat, and that heat transfers into the ground. So in this Pleistocene park, this park that they've been doing this experiment in Siberia for a while, they put in a couple hundred animals that aren't mammoths, right? They put in ox and reindeer and things like that, and they're knocking trees over with the tractors. And once they knock trees over and they simulate a mammoth knocking the trees and shrubs over, the fleet grazers are able to keep the vegetation from regrowing. So when the vegetation doesn't regrow, you get all this grassland, and the grassland has snowpack. The snowpack gets stomped, so there's no insulation. It reflects more light. It's like 3 or 4 different processes that make the ground, I think on average, it's like 8 degrees colder. So it keeps things more frozen. So once we removed all the megafauna from the Arctic, through hunting or maybe other means, regardless, once they were removed, the Arctic got warmer. The Siberia and Alaska got warmer, and so slowly we're getting more and more carbon emissions from up there. But by putting these animals back, and I just love the idea of going up to the Arctic and it looking like the African savanna, right, with all of these incredible animals. But by putting these animals back, it in theory will make the Arctic colder, slow down the melting of the permafrost, which will in turn trap the carbon for longer in the ground.
Well, that's it.
I'm not entirely sure how he wants to do that by... I'm not sure I follow, because, okay, you put some mammoths back. How is going to be how is the resupply of one species and the changes in the ecosystem it does, how is it going to have a big effect on global warming or making the Arctic colder? I'm not entirely... Yeah, I think he's a little bit too optimistic there. And I think maybe also nowadays, people have discovered that nowadays you can sell many things and get a lot of funding if you say it's against it's global warming. It's a lot of scam, is what it is. So I'm not sure. I mean, he says that it doesn't make much sense to me, and it's just some gobbledygook to get some extra funding.
Mm-hmm.
I mean, he may have a point. There may be a little bit of a temperature change when you put organisms there. But I mean, what people estimate happened was that after Genghis Khan was finished with his ride to the Asian continent, then that global temperature would cool a little bit because he had killed so many people. How is I believe that, but I mean, how is repopulating the Arctic with mammals going to be, or with mammoths going to lead to a similar change, a significant change of temperature? I'm not entirely sure about that. I think it's an interesting idea. It's worthwhile to try out. Yeah, why not? Try putting mammoths back. And yeah, I think it's cool to study. Why not? But I'm not convinced that it's really for a bigger purpose to combat global warming or whatnot. That just might be his way to get more funding.
Could be. Just out of curiosity, I pulled up the website here. Let's see. I think the company is called Colossal. There we go. Colossal. If you're curious, check this out later. But yeah, any final thoughts on bringing back woolly mammoths? You know, even if it's not for climate change, how do you feel about the situation?
I think it's cool. I mean, if they want to do that, in the Arctic, that's fine with me, right? It's far away, so what should happen? And we can study how the reintroduction of organisms into ecosystem, what actually will happen to the ecosystem, right? And it's also not as hysterical, the reaction. I mean, there's not many emotions. It's not the same as with the development of the COVID vaccine, where there's a lot of emotions involved and people are afraid of dying, and then they push a vaccine forward without testing it further. You can take your time to really observe what will happen in the Arctic once you reintroduce such a vast species as the mammoth. And so you can study it, and yeah, I agree with the guy. It would be cool to see the Arctic as like a tundra again, that like is like warm or something. But I don't know whether that will really happen. But it sure would be cool to have mammoths there. Yeah, why not? And there's going to be some interesting changes to the entire ecosystem just because you introduce a new species. I mean, there's a lot of things that can go wrong, but there's also a lot of things that can go right. Yeah, I'll be on board with that. I think that would be cool. I would have no ethical problems to working with them, for them. So yeah.
All right. Cool. So let's see. On a scale of 1 to 10, how cool would you think this would be to see mammoths again?
Pretty much an 8. Yeah.
On a scale of 1 to 10, how successful do you think this will be? Like, do you think they will succeed in bringing back a woolly mammoth? 1 to 10.
Yeah, I would give it slightly back than average. I think people tend to be more optimistic than possible, but I'd give it a 60% chance. 6 out of 10.
Okay. Do you think on a scale of 1 to 10, how dangerous could this be?
A 3 out of 10.
Okay.
It's the Arctic. It's like far away.
Okay. Scale of 1 to 10, with 1 being non-ethical to 10 being very ethical. What number do you give this experiment?
10 being very ethical?
Yes, like within, like 10 is like everything's cool, everything's above board. 1 is like we're Nazis experimenting on babies, basically.
Okay, 8 out of 10.
Okay, damn. All right. Okay. And then final question, did you watch Jurassic Park?
Oh yeah, I even watched the last part.
Haha. So, did you
And
like
the animated series.
Oh, I guess you like Jurassic Park then, it seems.
I have to say this: Jurassic Park is one of the best soundtracks ever. I mean, they really got that right, and the rest is iconic. The unfortunate thing for Jurassic Park is just that it has come out in the time when it was it kind of a transitional time, right? It didn't exactly fall into the big blockbuster, and sometimes in the they had these movies like Troy and also Lord of the Rings, where they basically had like a huge settings, and then watching movies became very spectacular, and I think Jurassic Park fell into kind of like a strange. I think in the 90s everything was kind of changing, and so Jurassic Park could have had a more significant cultural impact than I feel it has. I think more people should know. Jurassic Park should be more iconic than it is thought to be. I think, but yeah, I like it.
Why do you think it's iconic? Why is it such a big deal?
Jurassic Park because a lot of things that Jurassic Park talked about are actually possible in principle. I mean, Jurassic Park, if you read the book, which is called Dino Park by Michael Crichton, they are talking about sequencing DNA in like a couple hours. And back in the 90s, let me tell you, that was utopian. Sequencing DNA, it took them 10 years or so for the Human Genome Project. It took them forever to get the whole DNA sequence. Craig Venter, the Human Genome Project was a publicly sponsored project, and Craig Venter did it with Celera, which was company, and Craig Venter did it a little quicker, but not by much. And they needed like 5 to 10 years to sequence the entire human genome. Today, you can sequence a personalized genome in 5 hours, the entire genome. And Jurassic Park talked about that ability back then in the 90s. It seemed utopian, but it's absolutely possible now. So that was prophetic, and the ability to put dinosaurs back. Yeah, man. I mean, they put mammals back. Now, in theory, if they just basically only the scarcity of dinosaur DNA prevents them from bringing them back. That is basically it. There's probably more there's more infrastructural challenges to bring back dinosaurs, but those challenges can be solved, right?
Well, with
Those
Jurassic Park, like basically it was like like it all seemed cool in the beginning until the dinosaurs escaped and then they started eating people and then you know what I mean? Then the raptors are super intelligent. So I mean like it seems like the movie in general was like this is not good, right? So when you say that Jurassic Park was iconic, do you agree with that idea that it was not good? And then was it iconic in the sense that like is the fact that these were creating monsters like part of it? Like, you know what I mean? Like how do you I guess you could easily see the connection here where they're literally calling it Pleistocene Park, right? You know, I don't want that to happen. I don't want there to be like, you know, like I don't want to be like fearing for my life. So,
right.
You know, I guess how do you?
Yeah, maybe I revised my ethical score from 8 to 10 to 7 out of 10. But yeah, seriously, I think because it also gives backing to people who think it would be cool to get dinosaurs back, right? And that of course would be not desirable. I don't think there's any doubt about that. That of course, I mean that's already in the that is not a good, that having a dinosaur, having a Jurassic Park is basically, you already know from the title that that's probably not a good idea, right? To bring back dinosaurs like that. If you bring back mammoth, I don't see how mammoth will actually be dangerous. If you bring back the saber-toothed tiger, that may and you put it into into Siberia, that could be a problem because those saber-toothed tigers could adapt to higher temperatures and then just start migrating from Siberia into Russia proper and then into the rest of the world, right? So that's that's going to be a problem. And by the way, I mean, could always happen that somebody accidentally, like somebody thinks it's a good idea to bring the baby mammoth back into the you know, back into Texas, and then all of a sudden you have mammoths in Texas. I mean, it's probably difficult to bring mammoths to Texas because, as the name implies, they are mammoths, so they can't be hidden so easily. But in principle, once they're out there, somebody could just spread them around the world, right? That possibility is there, or they
Right.
can actually get out their habitat. I just think that mammoths itself are just not, you know. I mean, elephants could also escape their breeding grounds. You're just not worried about elephants that are around in your neighborhood. You are worried about tigers escaping from the local zoo. So if they bring back the saber-toothed tiger, you know, that might be not so nice. But then again, you know, humans are pretty good, and that was Scott Adams once said, we are pretty good to react to slow unfolding disasters. So if we have enough time and we can basically find ways to deal with it, even if a dinosaur if dinosaurs were alive and well and were making their way back to like other parts of the world, we would have ways to deal with them because they don't suddenly appear somewhere, right? Even if they suddenly appear somewhere, let's say somebody brings a like a T-Rex into Manhattan, that would be one T-Rex, right? It would basically kill some people, and then people would be able to kill the T-Rex, right? It's not that all of a sudden, it's not that T-Rex, you don't have all of a sudden 24 T-Rexes beamed into the center of Manhattan. That just doesn't happen, right? So I'm not too worried about that, although there is reason to be concerned, of course.
Okay. Fair enough. Any final thoughts on this video? On Forrest Galante, on, you know, mammoths and Colossal, this company?
It's interesting. I didn't really. I knew they were trying to clone mammoth. I didn't really know about it before. I didn't really know about the details. Now I know a little bit more. I can look more into that. The claims with global warming, climate change, that seems to be far-fetched, but the rest is pretty cool.
Awesome. Did you expect me to come at you with this video today?
No, I didn't. I was entirely open to whatever video you had. That was interesting.
Cool. Yeah, I thought you'd be like the perfect person, you know, to answer this.
Thank you.
Awesome. All right,
Yeah.
cool. I think we're good for